Good afternoon, Penn Staters. Thanks for joining us. I'm Ryan Jones, Interim Executive Officer for the Penn State Alumni Association. I'd like to welcome everybody to today's virtual speaker session titled Animal Microbiomes and One Health. Live closed captions are available. You can access them by clicking the closed caption button at the bottom of the Zoom video window and then clicking Show Subtitle. You may also customize your caption view by clicking the stream text link posted in the chat. And we're live streaming today's presentation, which will be archived and available on our website. Today's guest is Erika Ganda, an assistant professor in Penn State's Department of Animal Science, and she's focused on infectious diseases of humans and animals. With a background in veterinary medicine, her work addresses foodborne diseases, antimicrobial resistance, and animal-based food safety. Additionally, she values data analysis and visualization and collaborates across disciplines, mentoring students and engaging in international research. Today, she'll explore how microbes in animals, people, and the environment are interconnected, a concept known as One Health. Professor Ganda, thank you so much for joining us.
Thank you so much for the invite. Can everybody hear me okay? Yes? Okay. So I'm going to share my slides. Everybody good? You can see a single slide? Great. So I'm going to jump right into it. I have a little bit of my background and what I do at Penn State baked into the presentation, as well as I'm going to share a few stories on work that I have done in my previous research life and also things that we are doing here at Penn State. My name is Dr. Erika Ganda. I am an assistant professor of food animal microbiomes in the Animal Science Department, co-hired by the Huck Institutes of the Life Sciences and a member of the One Health Microbiome Center. So starting with the big picture, I like to take the elephant out of the room and tell you where the accent is coming from. So I am originally from Brazil and I graduated with my vet degree in 2012. And as part of the last 6 months of vet school, you need to do a practicum. And that led me to the University of Florida, where I was first introduced to animal research and large dairy farming systems. I did a lot of dairy farm work while a vet student. So I was really excited to learn how the systems operated in the US. And there, I first learned about the word microbiome, which took me to Cornell University, where I first did a PhD, then a postdoc, which I'm going to share a little bit about, focusing on the milk microbiome. And since July 2019, I started my lab here at Penn State in the Department of Animal Science. One thing that is really cool about these three institutions that I've had the privilege to work in is that they are all land grants. And the land grant mission is a commitment to education, research, and outreach that benefits society. It's something really special that the United States has. And our goal is to solve real-world problems through science. It was created by the Morrill Act in 1862, supports scientific discovery, and bridges universities through extension program. And as a Penn State have a fantastic bridge between researchers and extension and the general public through initiatives like this webinar, where I'm trying to bring some of my research to folks who are alumni. So I would like to also introduce you to the concept of One Health. You probably have heard about it, but if not, think about it this way. For you to be healthy, you need to have healthy air to breathe. You need to have healthy food to eat. And for you to have eaten your steak or your eggs this morning, you need a healthy crops to be able to feed those chickens or those cows and so forth. But the way that I look at that is that the glue that keeps all this health together is the flow of microbes that flows through and around us. For example, every fall, we would, if it wasn't for the microbes that live in soil and degrade the leaves, we would be swimming in trash. So microbes do extremely important ecosystem services to us, and this is the lens that I view the world. Speaking of the world population is expected to reach 10 billion by 2060, and we are not getting any more land. In fact, we're losing a lot of our agricultural land. So we have a big challenge in terms of animal science and food production on how to properly feed all these people. And that brings me to my laboratory, where we focus on animal microbiomes with a One Health perspective. And our goal is to develop microbiome-based solutions for the use in agricultural industry. I like to say that I don't do any of the work. I mainly beg for money and fight bureaucracy. The real stars of my team are the ones depicted here who do all the hard work. And one thing that I'd like to point out is that we are very diverse in terms of what we work with. Underneath each student, you're going to see little icons of animals and molecules. So we really try to study animal microbiomes from a perspective in which the student is interested in. Another thing that's really special about Penn State and that brought me here is the existence of the One Health Microbiome Center, who has been highlighted in this webinar series before. So this is a picture that truly shows our interdisciplinarity. And in this picture, you can see folks that work in food science and computer science and plant pathology and biology and plant science. So it's a really interdisciplinary group. And we have grown a lot and are doing really cool things around the microbiome. But I'm talking about microbiome, and I would like to actually define the word. So what is the microbiome? Is the collection of microorganisms such as bacteria, fungi, viruses that live in and around us, and they have been directly linked to many things. We usually think about infectious disease, but our microbiome is actually very fundamental for training our immune system and combating disease as well. One thing that I'd like to highlight is that a lot of we know in terms of microbiomes has to do with bacteria because we've known more about bacteria. We have been able to grow them in the lab and manipulate them better. But we can't forget about the other aspects of the microbiome, such as the fungi, the viruses, the protozoa that we have. And there is still a lot to be learned. So talking about bacterial land, until the advent of microbiome work, a lot of the things that we would think about when you thought about bacteria is infection. It's something bad and you need to try to get rid of it. But in fact, there are also many good things that bacteria do for us. And one example is bacteria can be used to actually train the offspring's immune system. So this illustration shows how we have evolved as mammals to have our immune system go inside the mom's gut and then transport little bits of this bacteria and dump that into the milk that then teaches the offspring's immune system, hey, this is good microbes that came from your mom and you should be okay with it and learn how to tolerate them. On the other side, we have a lot of evidence of negative effects of microbiome and imbalance. Infections are one of them. Infectious disease is the first thing that comes to mind, but there are a lot of research showing that imbalances on the microbiome have been linked to anxiety, depression, chronic inflammation, diabetes, things like cancer that we've never thought about. We are starting to learn that depending on the kinds of microbes you have in your gut, you might have cancer treatments that are more toxic or less effective, depending on the bacteria that are living inside of you. And now we need to take that into account when choosing cancer therapies. So I gave a little bit of a broad overview of the microbiome, but focusing more on my laboratory. What we study here is food production, antimicrobial resistance, alternatives to antibiotics, zoonotic diseases, and public health. These are the main topics of interest of our lab. And today I'm going to share you a few stories on a few different species and work that we've been doing in our lab. So starting with cows, I've worked with dairy most of my life, and I started my journey working and studying the milk microbiome from the milk producer side. So I studied a disease called bovine mastitis. It's the inflammation of the mammary gland, and it affects a quarter of the US dairy herd at least once in their lactation. Actually, it affects more. And from this quarter of 9.2 million cows, 22% of them get treated with some kind of antibiotic. And when you look into the antibiotics that are being used in dairy farms, 65% of these antibiotics are cephalosporins or third generation cephalosporins, which are considered critically important antibiotics for human health. So one of the problems that we were trying to look at is the use of third generation cephalosporins and mastitis. So when you treat a cow, um, some people have mentioned that in gram-negative bacterial infections, for example, those caused by E. coli, if you treat third generation cephalosporins, you could increase bacteriological cure. On the other hand, when you decide to give that medication to the cow, that there are a lot of costs that are associated with it, and also the problem with the rise of antibiotic resistance. So, what is antimicrobial resistance or antibiotic resistance? Is when the drugs that we have developed to target and kill specific kinds of bacteria no longer work. And there are many ways that bacteria can achieve this, and I'm going to share a little bit with you. And this is one of the biggest problems facing humanity. So this is a report from the United Nations saying that we really need to strengthen our One Health response to antimicrobial resistance. And here you can see a timeline. So 1910 to 2025 in green, we see when we first deployed the different kinds of antibiotics. And then in red, you can see when resistance arose. So antimicrobial resistance is a natural phenomenon. That's how bacteria have evolved to fight and compete with each other. So it's not something new and it's not something that's going to go away. It's a matter of preserving the effectiveness of certain drugs. And I'm going to give you some examples further in this presentation. But it is indeed a huge problem. And it's expected to be killing as many people as cancer by 2050. And this is a research article that was recently published that predicts that by 2050, mortality will rise by as much as 70%, and this mortality will be associated with antibiotic resistance in people that are age 70 and older. This is another little graph to put into perspective. We have antimicrobial resistance deaths around 2020 and what we expect to see by 2050. So we will really be seeing lots more deaths associated with bacterial infections that we cannot treat with the drugs that we have available. Another fun fact or not so fun fact, a lot of cancer patients that end up succumbing to disease are not really dying from their cancer, but from hospital-acquired infections that cannot be treated with antibiotics. So I told you that stopped working, but like how do bacteria and fungi fight back? So bacteria are extremely clever and they can do many different things to make the drugs that we're trying to use to kill them not work. So they can develop new processes. If I have a drug that targets the cell membrane, it can change the way that membrane works, or they can create molecules that chop up the antibiotic itself. They can hide and restrict access to the drug, or they can actually pump out any drug molecule that we expose them to. And one of the things that bacteria are extremely cool about is that they can change genetic elements. One way that I like to say this is imagine you're walking by somebody and you really like their hair and their hairstyle, and it's like you could grab a chunk of their hair and put on your head, and the next day you have the exact same kind of hair. That's what bacteria can do in terms of antibiotic resistance and gene and mobile genetic elements. These are a few of the ways bacteria can spread and give their hair color to one another. In the process called transduction, we use bacteriophages that then incorporate these genes that confer resistance for either those enzymes or pumping those drugs out. We can have bacteria to bacteria transmission, and bacteria can actually just pick up naked DNA from the environment. So how do we track this? It's a very simple process. So here I'm showing a plate where we grew a lot of bacteria and we have a lot of little dots. These dots have different antibiotics with different concentrations, and we measure how much growth or inhibition of bacteria we have for each drug. And if you have an antibiotic like this, meaning the bacteria can grow really close to it, means that that bacteria is resistant to that antibiotic. Now we have more high throughput methods. So here are some examples of what are used in current diagnostic tests. And with the advent of sequencing technologies, now we're able to actually look at the genome of this bacteria and be able to track resistance that way. And one of the things that I wanted to highlight, which in my opinion is one of the biggest success stories of One Health, is the National Antimicrobial Resistance Monitoring System. This is a United States program that is a partnership between FDA, CDC, and USDA, where they track enteric bacteria, look at antibiotic resistance, and later on in the presentation, I'm going to give you some examples of how this has actually changed how we produce animals to safeguard human health. So going back to the dairy value chain and my journey, for my postdoc, I studied the milk microbiome from a human health perspective in a project that was in partnership with IBM. And what we did there is, can we use artificial intelligence and the information on the microbes that are in the milk to be able to tell if this milk came from a farm I didn't expect it to come from, or it has a potential problem. And we published this work last year. In terms of what's going on in my lab, another disease that we like to study is called clinical metritis. So that's the inflammation of the uterus of a cow after she gives birth, and it can also affect up to 25% of cows. It is very costly, and it's another cause of use of antibiotics in dairy farms. So Janine is a really talented student who is now faculty at NC State, and one of her studies was, can we find an alternative to antibiotics to treat these cows? And she published this paper last year, and we were able to show that just by giving a concentrated sugar solution, this was a study in partnership with Dr. Adrian Barragan from Penn State Extension, we were able to show that there was no difference in clinical cure. So with that, we can show that we found a potential alternative treatment with no negative impact in health. We can improve antimicrobial stewardship and potentially decrease costs. And this is important, especially for things like organic production, where antibiotic use is not an option. Speaking of antimicrobial resistance and One Health, I would like to share a couple stories about my favorite bacteria. Her name is Salmonella Dublin. And the reason I like Salmonella Dublin is because she's a poster child of a One Health challenge. It is bovine adapted. When it infects humans, it causes severe disease, and it can survive in the environment for a long time. So I already told you the why, but the other why is, if you've heard of Typhoid Mary before, I hope most of you have heard of Typhoid Mary. She was a cook in the 1900s that was associated with a lot of deaths, and she was a healthy carrier of Salmonella typhi. So whenever she would cook food to people, people would contract Salmonella typhi or typhoid fever and die. Well, Salmonella Dublin is like Salmonella typhi, but for cows. So you can have a Typhoid Mary dairy cow in your farm, meaning that she looks completely healthy, but she's just spreading the bacteria whenever she's stressed out. And this has zoonotic potential because we have lots of research showing that these bacteria can indeed infect humans, and when it does infect humans, it's severe disease. So now I'm showing some data from another really talented student, Sophia, who used the NCBI pathogen isolate browser and looked at the genomes available for this bacteria from 2002 to 2022. And what we see is that there are a lot of genomes that are being sequenced. This is not because we see more of the bacteria, it's because we have the technology now. And through programs like NARMS, we can now collect and sequence these bacteria, and we can use those data. So it is a One Health challenge because it causes invasive disease. It can be subclinical carriers or your Typhoid Mary, and can also infect humans. And this paper shows, we're making a comparison between Salmonella Dublin and all other Salmonella. And it was looking at humans that had contracted Salmonellosis. And Salmonella Dublin has a significant higher death and hospitalization rate when compared to all other Salmonella. And most of the Salmonella Dublin are being isolated from blood. So that means that it's very invasive and severe disease. It's also heavily antibiotics resistance. So again, we have Salmonella doubling in one column, or other Salmonella. And Penn susceptible means if I treat this patient with this Salmonella with any drug, it's gonna be a successful cure. That bacteria is gonna die because it's susceptible to the drug. Well, Salmonella doubling, about 31% of them are highly antibiotic resistant as compared to 2% of all other Salmonella. And I can show you a little bit of data as of why. So this translates to a lot of economic impact. If you have Salmonella in your farm, it can cost anywhere from 49 to 300 euro. And this is kind of an old study, but that's all the estimates that we have. And one of the things that we sought to understand was what we know about Salmonella doubling in cattle and humans in the United States. So looking at the literature in 2018, we saw that there were lots of 46 out of 5,000 farms in New York State were positive for this pathogen. And I wondered once I started at Penn State, what happened to Pennsylvania? Like, do we have any data from here since we share a border and there's a lot of dairy that gets traded between both states? And I found this manuscript that showed that Salmonella doubling that was highly drug resistant was very similar between beef isolates. So here we have our beef isolates and here we have human isolates, showing that the bacteria were really similar. The way to look at this graph is imagine it's a family tree. So these would be kind of brothers. And when you look at these bacteria on a global perspective, I have color-coded the different continents here. And again, this is a family tree of bacteria. So things that are closer together are more relatively closely related, and things that are further apart are more different from one another. And what we can see is that all the isolates from North America pretty much fell on the same cluster. And they mostly carried this plasmid. Remember the ways that we can change antimicrobial resistance? Plasmids are one of them. They mostly carry this plasmid, and they mostly had all these resistance genes. So the dark colors here mean presence, and the light colors means absence of the different genes and different plasmids. But since 2014, we had this survey on animal health that is done by the USDA, and it has shown that Salmonella doubling was present in the United States, about 52% of samples from the West, and it's kind of making its way east. So it is an important thing to think about in Pennsylvania. And up to 40% of the large dairy herds have been shown to be positive for this bacteria, which tracks with its epidemiology. But this is not a new problem in Pennsylvania. I found a study published in 1999 that had described 35 outbreaks in counties bordering New York and Pennsylvania, showing that this is a very atypical salmonellosis for this region because this is very invasive disease. So the problem is in Pennsylvania. We're trying to tackle it in different ways. So I mentioned about the National Antimicrobial Resistance Monitoring System, and that's how we gather the data that I'm going to show you. It is a really cool multi-agency partnership where we have surveillance for enteric bacteria from humans, animals, and food products. So the CDC gets every bacteria that comes from, every salmonella that comes from a human. The CDC takes care of it, sequences, and sends it up to the database. For animals, we have the Animal Inspection Service, the Food Safety and Inspection Service, and also the Agricultural Research Service are contributing to animal isolates. And then in food productions, we have state health departments and universities that are going into the grocery store and buying meat and trying to test and see if they can isolate bacteria from it. This is another way of how all these different things can be integrated and bring to actual change. So what we wanted to do is leverage this surveillance program that the United States has, and that's very unique, and try to learn more about Salmonella doubling. So we can get the bacterial isolate, extract its DNA, and perform sequencing. And when we compare those data, we can start seeing the relationships between bacteria, as well as relationships between people and animals that were getting sick with this bacteria. This is another depiction on a lot of alphabet soup here with the different agencies, but is really to show that this is a true One Health program that's integrated in many different levels. So a couple of examples on how this program has created change. For example, Campylobacter, we have through the NARMS program have been surveilling this Campylobacter, and they saw rising fluoroquinolone resistance until the early 2000s. And then once they saw a rise in Campylobacter resistance from retail chicken, FDA then passed resolution to ban this specific drug in poultry to safeguard human health. This is another example on multi-drug resistance in salmonella in poultry, and there was a major outbreak, and now cephalosporins are not allowed to be used for treatment in poultry, again, to safeguard human health. So these are extremely rich data sets that we can study these genomes of these bacteria to inform policy and protect public health. So what we wanted to do for this study from Sophia is to take this biosurveillance infrastructure and evaluate genomic characteristics from Salmonella dublin. This is the graph showing how many isolates we had from each HHS region. And what you're going to see here is if an isolate came from a clinical case of a cow, it's purple. If it's a clinical case from a sick human, it's going to be blue. And if it came from a food or environment, it's going to be teal. And one thing that we can see really easily is that depending on the region that the salmonellas are being sequenced, it makes a lot of sense. We have a lot of dairy production in these areas, so we have more dairy associated and cattle associated isolates being sequenced here. And what we see is that there is a majority of cattle-associated isolates, but we still see lots of human and cattle disease. So we went and we tried to study the genomes and try to ask the question, how related are these? Like, are the bacteria that are causing disease in animals different from the ones that are causing disease in humans? And when you compared the pairwise comparisons, we could see that they were identical 729 bacteria that came from carcasses and milk and meat were identical. In humans, there were 166 that were identical, and in bovine, there were 80 that were identical. However, the cutoff that we use, that CDC uses to call a cluster of isolates, it's 20 core genome SNPs. So this is, you imagine all the letters in a bacterial genome, the APGCs. If you compare the entire genome, these bacteria, these 80 bacteria, had 0 letters different. And now when we do foodborne investigations, we consider closely related bacteria that have up to 20 letters different within their genome. And when you start looking at this, you can start seeing this is still with 0 letters difference, isolates from meat and isolates from humans, and the same thing with bovine. So this is a true One Health scenario where you see bacteria that's causing disease in bovines, that's present in food, and that's present in human, being transmitted across all 3 different domains. And when you use that cutoff of 20 that I just mentioned, that's what we are using per CDC guidelines for determining if bacteria are closely related. 72% of all pairwise comparisons we did were substantially similar or had less than 20 letters different in their core genome. But we also like to not just look into the computer side of things. We like to get our hands on the ground as well. And I started working on this and started making flyers and saying, hey, we're working on Salmonella dublin. This is a problem because it manifests differently. And I had a farmer from Pennsylvania call me and say, hey, last year we got this in my herd and 46% of my baby cows died within a month. Can you help me? And in fact, there are some herds that can lose all their calves if they don't get a good grasp on the disease early on. So we took on the challenge. We have been helping this farm and trying to identify and sample where this bacteria is hiding. And another thing that I like to highlight is that it takes a village to be able to do these kinds of work. This is Dr. Cantor from Penn State Extension. She studies cattle health. And I like to show this particular picture here. This lady's Dr. Elizabeth Ransom. She's a sociologist that has worked with us on antimicrobial resistance as well. And we get to this problem needs to be tackled with a multidisciplinary perspective. You can't fix a disease issue if you can't properly address behavior change. You can't fix a disease issue if you can't properly diagnose the disease. So we have a very multidisciplinary team working on this. So once I find the bacteria, the cows can look at me and say, cool, you found the bacteria, but everything is antibiotic resistant. So what are you going to do about it? And another project that I'd like to highlight is on antimicrobial resistance and how can we detect what kinds of resistance are present. So the study is led by Samantha, another really talented student. And basically, she's looking at the bacteria and trying to find the genes. The state of the art now, if we get a sample from a cow and try to extract the DNA to find what antimicrobial resistance genes are there, it will cost us about $250 per sample, and less than 0.1% of the data you paid for is going to actually be the gene you're interested in. So what we did is, this is the kind of graph that you would be making your decisions on this very little sliver of color data, and everything here is irrelevant to your question. So we did some playing around with some molecular biology and used a real-time sequencer, and now we're able to only pay for the antimicrobial resistance data that we are interested in and can start using this tool to make decisions at the farm level. So now I'm going to shift gears and I'm going to talk about a different species and highlight some of the poultry studies that we have going on. I have mainly a dairy background, but there are fantastic poultry faculty and experts and facilities at Penn State, and I wanted to leverage that. And we study the microbiome as a component of poultry health. So this study is led by Anna, another really talented student in the lab. And why do we care about poultry? Because global meat production and consumption is increasing, and poultry is one of the most rising demands. And since 1940, poultry producers used to feed antibiotics at low levels for growth promotion. And that used to improve feed efficiency, increase growth rate, decrease mortality, and enhance intestinal health. However, global legislation, and in the United States since 2017, the Veterinary Feed Directive has put lots of restrictions in terms of which drugs and how you could feed these animals. And this is leading to a worldwide need for antibiotic alternatives. This is a report from the FDA showing that, yes, this regulation worked. So here we had the annual total sales for antibiotics before that 2017. The blue is you could just walk up to the store and buy, and the green is you need a prescription or a veterinary oversight. So it's another way that policy is being used to help consumer food safety and demand. This is another depiction of how we are decreasing the amount of antibiotics that are being sold per year. It's specific. This one are specifically for poultry, and most of the antibiotics that are being used are tetracyclines, which are not that used in human medicine. So even though we still use a lot, they're not the most critical antibiotics that we need for human health. And another thing is consumer demand. A lot of folks go to the grocery store and see no antibiotic ever. So consumers want to buy this. And the farmers didn't want to feed antibiotics because they liked to. It was a useful tool in their toolbox to produce healthy animals and maintain profitability. So that led to a lot of different products in the market to try to fill this void, such as probiotics, postbiotics, phytotherapeutics. And one question that we sought to address is how do these compare to the antibiotics in terms of growth promotion and antibiotic resistance? So with that, we hope to provide unbiased science-based information for poultry producers. How do these different alternatives to antibiotics act in the poultry microbiome to help respond to consumer demand and maintain profitability? So here I'm just sharing some cute pictures of Anna with her day-old chicks. and a few pictures of the group. It's a lot of hard work. So this was a 3,000 bird trial that we ran at Penn State, amazing facilities, and everybody needs to chip in, myself included, when we had to weigh birds. So a lot of fun. Anna is currently working on that data right now. And I also wanted to share some things that I work beyond animal agriculture. I am a veterinarian by training. I really care about agriculture. Farmers work really hard to put food on our table every day. And one of the things that I like to highlight is people really like to blame the chicken that they ate or the beef that they ate, but nobody's really talking about the potential role that their dogs could be playing on disease. So I don't know about you, but I don't let a cow jump on my bed every morning and snuggle with me, but I let my dog, and there is very little surveillance in terms of the role of antibiotic resistant salmonella in pets and humans. So again, with publicly available data, 59 million households in the United States own a dog or a cat. And this leads to an average of $1,800 spending per pet owner per year. So it's a huge market. And what we did was very similar to the Salmonella doubling in humans. In bovine story, we went into the data set. We took publicly available data, and we found a quite small subset of like 77 humans and 87 dogs, and we compared their genomes. And with that small data set, we found two clusters of Salmonella enteritidis that were 99.996 identical between dogs and humans. Now, this is surveillance data. I can't tell you that that bacteria came from the dog to the human or from the human to the dog, but I can tell you that the dog and the human had this bacteria and they were pretty much identical. And we also saw another cluster for Salmonella infantis. And there is a lot of evidence for this. So these are snippets from the CDC website on outbreaks in humans that were associated with pets. So pet treats, if you feed your dog a specific pet treat that is contaminated with bacteria, you could get sick with that. So just highlighting that we shouldn't really only be looking at farm animals, but actually all the animals in a One Health perspective. Another thing I wanted to highlight today beyond agricultural microbiomes is my course, Animal Science 456. And this is a undergraduate course that I developed once I joined Penn State to expose undergraduate students in the College of Ag to microbiomes. And this is a very hands-on course where they get to experience, we visit all the different core facilities and they learn about all the different technologies that are used to study this field. They actually get their own data set and they analyze their own data. And by the end of the semester, they have a mini symposium where they explain what they found through the data they analyze over the semester to their colleagues. And I also like to have fun. I believe that once you're having fun and you're learning something, you're going to retain more. So here are some examples of things that we do to learn microbiome. We have a game board. This is a microbial community that they keep kind of like a pet over the course of the semester, and they watch how the microbes grow and the colors change. And at the end of the semester, they bring those microbial pets and they compare to one another, and it's a lot of fun. And last but not least, being from Brazil, I care a lot about global health and public health and international work. So I wanted to highlight a couple works that we've done in collaboration with Brazil and now initiatives in Kenya. So we have this 2020 study where we studied a ecological disaster. So there was this mining dam that ruptured and contaminated waterways with iron ore waste that was heavily contaminated with heavy metals. And we asked a simple question: is there any association in the bacteria within those animals drinking their water or not? Again, I can't say for sure that's what happened. I saw associations that exposure to heavy metal contaminated water was associated with more antimicrobial resistance genes in dairy cattle that were drinking their water in Brazil, because there is this phenomenon called co-resistance or co-selection, where you put something and you expose the bacteria to a higher copper, for example. But in those mobile genetic elements that they can transfer between them, it can confer resistance to copper, but it can also confer resistance to a different drug. So here I'm just sharing some pictures from visits and workshops that I have been given there. I have the pleasure to work with very talented folks. And this is the university where I did my vet school. So it was really fun to go and be able to see my mentors and I'll work together with them. But most importantly, I care a lot about bringing folks from that have less resources to Penn State and be able to have that connection here and share all the amazing facilities we have at Penn State and the expertise we have as well. And then in Kenya, this is a kind of recent initiative. This university in Kenya contacted Penn State and said, hey, we want to work together. How can we do something? We did lots of meetings trying to find out where our expertise is overlapped. And that culminated on a trip and a memorandum of understanding that was signed in 2023 between the university, the county government, and Penn State, which basically said, yes, we are committed to this and we're going to work together. But I didn't want to go across the world just to take a picture and look pretty in a photo. I wanted to actually get something concrete done. So what I did is I packed up a lot of reagents and equipment, and I did capacity building. So we had a molecular biology training that was done there, some bioinformatics, and we are actually getting stuff done. So here are some examples of the data that we collected as part of that trip. And again, I care a lot about sharing the facilities that we have here. So this is my right hand, Frank Onyambu from Kenya. He was able to visit Penn State last year, and we continue to working together. And with that, I hope you enjoyed the wild ride. I'd like to acknowledge all my funding sources, our collaborators, and my fantastic lab group. And I'd like to thank you for your attention, and I'll be happy to answer any questions.
Erika, thank you so much. That was you ran through a lot there. It's a ton of information, and the work is phenomenal. I've had a little bit of a chance to get familiar with this, my work with the Penn Stater magazine and some of the coverage we've done. But there's so much there, and the impact, as you got at, is really massive. We do have a few questions. You talked a bit about obviously your path to Penn State, but can you say a bit more just about what makes Penn State? So much of the nature of the One Health work is collaborative by definition, right? It has to involve people from all different areas. What has made Penn State for you a place where that kind of collaboration can happen at a high level?
The existence of the Huck Institutes and all the institutes, the interdisciplinary institutes, is something that's really special to Penn State. And in a lot of fields, people are sometimes discouraged of going too far from their own area of expertise because they want investigators to build reputation on their expertise, and that's not necessarily the case at Penn State. We are valued for interdisciplinary work as well, and having people value the fact that I work across chickens and cows and dogs and with people in plant science and statistics. It's really fantastic, and I think kind of unique to Penn State. Um, the environment is really collegial. I absolutely love the people I work with. We have folks that are extremely successful and published in Science and Nature, and you can send them an email and say, hey, I have some questions. Would you help me think through this study? And they say absolutely, and give me an hour of their day and we'll talk through things. So that's what I think is absolutely fantastic. And we have all the facilities we want. I'd love to give you a tour of my lab. We have a new building in animal science, and I love where I am. We have all the equipment and expertise. And the One Health Microbiome Center that I'm heavily involved on is another reason why this kind of interdisciplinary research is so successful.
I would love to take you up on the tour one of these days. Absolutely. Um, can you talk about how your work, again, you highlighted a lot of the students you work with, the grad students in particular. How do you sort of balance that, and how does that fit in the teaching and mentoring with obviously the research that you're focused on? How do you balance that, and how does doing that kind of fit in with your philosophy and approach?
My philosophy to mentoring is I am a facilitator to help the students get where they want with their career. And that has led to kind of the scattered approach of why we're working in so many different species, because everything has a microbiome, and I'm a microbiome expert. So I work with my students to try to identify what their interests are, secure funding for those interests, and that way these students are highly motivated. They do fantastic work. They come to me with really good ideas. And that approach is different than handing a student a set project and you have to work on this project and this is what your PhD will be. So I try to marry those two approaches. Sometimes we have a project that needs to get done, but I also heavily encourage my students to what drives you? What are you interested in? Let's write a project and let's look for funding. Let's make it happen in a way that is something that you care about and that's impactful. A lot of open communication, collaboration. We run lab meetings that I have kind of a industry mentality where we have group accountability. So we share what we did last week, what we are doing this week, and what we need help with to really facilitate students helping each other and sharing experience and expertise, and that makes them develop much faster than if they were in their little bubble trying to troubleshoot an essay, for example.
Thank you for that. What sort of feedback, again, you do presentations like this, what do you hear?
You're muted.
Sorry. What sort of feedback, I'm wondering, do you hear from whether it's alumni who you've made presentations to, or just folks who are not in the sciences necessarily, but that the impact of this work is translating, that people are kind of get the importance of it and the significance of it?
I really like to show the NARMS examples and how regulation and surveillance have made real impact in safeguarding public health. Like the United States food supply is one of the safest in the world, and that's because of programs like that and experts like us in the mix. Things like the probiotic story and the dogs and their role in potential infectious disease story are also different ways that we can have impacts on daily lives of folks, even though we're just doing lots of supercomputer big data stuff.
And then we have a few questions from some of the folks who've been on the call. A general question about probiotics or prebiotics, particularly, I think those get suggested for a lot of folks when they're taking antibiotics, right? And again, we sort of hear it as like, okay, if that'll help me stay healthy. I guess, is that a suggested thing based on what your research shows? And also, can those sorts of things impact mood and behavior when you've got that going on?
Yes, there is a lot of research showing that the impact of bacteria can have in mood and behavior through the gut-brain axis, anxiety and depression. My take on probiotics is we're not dealing with one bacteria, we're dealing with a community of bacteria that's in our gut, and then you're trying to put an intruder in. And the effect of probiotics doesn't really rely only on that bacteria taking hold on your gut. So we have instances where given a postbiotic, which is you grow a lot of bacteria and then you kill everybody with heat, and then you dry that up and make that into a product, and then you take that product of dead bacteria and all the metabolites they made, and that itself can have a positive impact in health. For probiotics, that for you to see a benefit, you need that bacteria to take a hold on your gut. That can get a little bit trickier. And the analogy that I like to make is imagine you got invited last minute to a party, and then you get to this party, you open the door, and everybody was partying and talking, and then they look at you weird, and then they shut up. Do you want to be on that party? Not really. So that's kind of like the probiotic bacteria that's showing up to your gut party, and the environment that's there, it's really going to influence the success of that therapy. That said, if you're taking antibiotics don't discriminate. They kill all bacteria of that specific class that you're targeting, not only the bad ones, but the ones that are commensals. So if you're adding something back in, it's a good thing. Just if you take your probiotic, don't take it at the same time as your antibiotic. You have a gap between.
Gotcha. Okay. That is, and I like that analogy. That's very helpful. Couple of the questions from listeners. Someone's wondering if you or your lab had been impacted at all by recent cuts.
Directly impacted, not yet, thank God. Penn State has been very helpful in navigating these uncertain challenges, but we have been severely impacted from a what is the future going to look like perspective. A lot of my funding and like a lot of the surveillance funding comes through USDA and FDA, and all of that is up in the air. I have students that submitted fellowships last year that would help fund the rest of their doctorate program that we don't know. Are they going to have the funding? Are they not going to have the funding? We haven't heard anything. We're supposed to hear it by now. I have students that had contracts and they were supposed to join the US government upon graduation. And now we don't know if they have a job or not. So, yes, we have been severely impacted, not in terms of the money got taken away, but the uncertainty in the future is substantially impacting the lab.
Two more questions here, as I know we're getting close on time. One, obviously, you talked a bit about domesticated dogs, which so many of us are dog owners. The question is, what would be considered a normal microbiome for a dog? And is it different for dogs that might do sort of different activities, a show dog or a dog going through obedience training or a dog that you would bring in the field for hunting or something?
It's funny that you brought that up because we do so much work in mice, but actually dog microbiomes are much more similar to human microbiomes than mice microbiomes are. And there are some papers showing that like people that live with dogs in their household have more similar microbiomes to their dogs. So for that question, a healthy microbiome, it's not a one certain thing because it's specifically dependent on the geography and the kinds of activity and the kinds of diet. So like there is a healthy human microbiome, but the healthy human microbiome that lives in North America is going to look different from a healthy human microbiome that lives in Africa, for example, because they eat different food, they have different environments. So there, the overarching theme is that a diverse microbiome, a well-balanced microbiome is associated with health. I am not aware of studies that have specifically compared dogs that do different activities, but I'm aware of studies that looked at humans that do different activities. And yes, athletes have different microbiomes than people who are sedentary, so I would expect yes to see. But it's kind of a tricky thing because how do you control for the diet? Like for you to be able to really answer that question, you'd have to have dogs that are very similarly from a genetic background that are fed the exact same food, and then you make them do different things, and then you compare their microbiome. So that's kind of hard to answer directly.
That makes sense. And that actually was our last question. So we're running up on the hour here. I want to thank you so much for your time. It's great to be able to share this research with folks again. And I think you did a great job of conveying it in a way that people can kind of understand the practical applications, which obviously is a huge part of it. And I want to thank everybody for tuning in as well. I know you shared it in your, I think, last couple of slides, but as far as where people can get more information about your work, well, again, this is recorded, so people can go back and look at that. But is there kind of a quick way to send people to find out about your work more easily?
Gandalab.org or just Erika Ganda in the Penn State website, and you're gonna easily find me. I'm on Twitter, X, and LinkedIn. So yeah, I have several different ways to find
not
me.
hard to find.
Not hard to find.
Okay, great. Well, thank you again. It was really a terrific session. We really appreciate it. And for folks who want to access this event or any of our other virtual recorded events, in-person events upcoming, or things we've recorded from the past, it's alumni.psu.edu/events. Thanks again. And Erika, you know the cue. We are
Penn State.
Thanks. Thanks, everybody. Appreciate it.